And a happy Monday to all you micro friends. I thought we would go over the exam. I posted the study guide for the next exam. I'll get the exam out to you hopefully tomorrow. I'm still kind of tweaking on it, trying to make sure it matches up with kind of what we've been talking about. So here is exam 2. And this was on some of the life cycle and genetics and stuff. Just to go through it here, we're starting out with question number 1. Organisms require energy from outside their bodies. Heterotrophs get energy from organic compounds made by other organisms. And phototrophs get energy from the sun. Lithotrophs get energy from inorganic compounds. Let's see, question 2. Using pyruvic acid to make ATP by the process of respiration is the best answer. But if if the organism can discard used electrons, that is, it can dump the electrons onto an inorganic compound like oxygen to make water. Pyruvic acid is being used to make ATP by the process of respiration. If it's discarding electrons onto organic compounds, then that is what we define as fermentation, where you're using basically pyruvate as the final electron acceptor. All right, animals must use oxygen during aerobic respiration. You and I, we are obligate aerobes. So products of reactions with oxygen can be toxic in bacteria that cannot, not will not, but cannot detoxify them. These are going to be obligate anaerobes. So feedback inhibition is usually regulated by the concentration of the products of an enzyme catalyzed reaction. So as you start to generate more stuff, then that can begin to feed back on the system and cause it to slow up. Let's see. Glucose can be used to produce ATP during respiration, but glycolysis and Krebs cycle reaction intermediates can also be used to produce monomers such as amino acids to make proteins. That was question 5. All right, statement 6. Heat and many disinfectants denature proteins primarily, since enzymes are mostly proteins, and they're going to also disrupt the cell membrane. Soap does that too. Statement 7. The phenol coefficient is the measurement of the toxicity of the chemicals, and if it's greater than 1, it is a disinfectant, not an antiseptic. And disinfectants are pretty nasty. Let's see. Statement 8. Essential genes are located on the bacterial chromosome with optional plasmids found in many prokaryotes contains genes that might be useful. Let's see. So statement 9, often inefficient. Cells can import DNA directly from their environment by the process of transformation, that is, picking up a plasmid. The efficiency of DNA transfer is increased either when viruses, and that's transduction, or other bacteria, conjugation, guide the DNA into a recipient cell. Yeah. All right, multiple choice questions. Let's see how we do here. Starting with question 1. A great source of chemical energy from food, broken down during respiration to make ATP. Pretty much the only food there that is got all the carbons that you would want is glucose. Lactic acid and ethanol are broken down, but they don't have as much carbon as glucose does. So it's not a great source. They're sources. Catabolic reactions are using hydrolysis reactions. This is the breaking down reactions. Anabolics are the building ups. Enzymes are essential for life. They are reducing the energy of activation, A. Let's see, 4, feedback inhibition works by regulating enzyme activity when there is too much. Best answer would be C, product. Instead of using oxygen to remove electrons, anaerobic respiration is most likely to use something like non-oxygen inorganic elements, inorganic compounds. A is the best answer, iron, and nitrate, and sulfate. So instead of using glucose for chemical energy, lithotrophs are most likely to use something like some of these minerals, and they can actually break apart the compounds and extract energy from them. So the answer is C. Facultative anaerobes usually prefer to use aerobic respiration when possible instead of fermentation, and that's because fermentation is inefficient. 1 molecule of glucose in the presence of oxygen will give you 32, 36 ATP. 1 molecule of glucose in the absence of oxygen, that is carrying out fermentation, is going to give you 2 ATPs per glucose. So it's way better if you can use oxygen. Let's see, question 8. Um, Drying or high concentrations of salt or sugar are going to remove water from the cell through osmosis. The most useful method for controlling bacteria, viruses, and parasites in all cases is, well, there is no uniform thing. Steam heat is good for a variety of things, but it doesn't control things like bacterial spores. Filtration isn't going to do anything for viruses. UV light's pretty good, but again, spores are pretty tough, and different parasites might be resistant to it. No method works in all cases. So question 10, which process kills pathogens with heat but doesn't sterilize? That is, it is a blast of heat, so it's lowering the load but not actually sterilizing, and that'd be pasteurization. Question 11, UV irradiation is going to primarily work on solid surfaces because it can't get into things. It won't pass through stuff very well at all. Okay. 12. All forms of radiation kill primarily by destroying DNA. That is the best answer there. Filtration is effective for sterilizing air and liquids, but often allows viruses to pass through because they are very small. Unlike disinfectants, antiseptics are things that can be used to treat cuts and wounds rather than sterilizing. So antiseptics are not as nasty as disinfectants. Vocabulary term, whatever. Let's see. 15. Main purpose of using either disinfectants or antiseptics. They're not necessarily going to sterilize the surfaces. That's going to take some stuff that's a little bit more harsh, like sterilants. But they will reduce the number of potential pathogens. Is a better way of thinking about it. Mutations occurred in the production of what compound in the cells? Well, mutations are only going to be useful if they are changing nucleic acid sequence in DNA. So 17, every mutation causes, by definition, a mutation is a change in the genetic material. It may not have an effect, but it is a change in the genetic material. So that's A. 18, unlike eukaryotic chromosomes, chromosomes of prokaryotes are circles. Eukaryotes, you and I, we have linear chromosomes. They have a start and a stop. An R factor is likely to carry drug resistance genes from one bacterium and another in the process of conjugation. R factor is part of that conjugation business. Which of the following cannot enhance genetic variety in bacterial populations? Best answer would be sexual reproduction because there really isn't any sexual reproduction in a bacterium. Let's see, 21. The first appearance of any new traits, such as new drug resistance, is going to be caused by mutations. That is how it happens. So the other things are not actually causing. They may be selecting for, but they're not going to be causing. Plasmids promote the spread of drug resistance by moving between neighboring cells by the process of transformation. So moving plasmids from one cell to another, that is transformation. That's C. The pattern of passing genes from parents to the offspring, this is going to be vertical gene transfer. As opposed to like that. All right. Let's see. 24. An antibiotic that blocks the bacterial ribosome will kill these cells because they can't translate proteins. Ribosomes are the structures in which the RNA lays on, and that's how you actually are able to synthesize proteins from RNA. So B is the best answer there. Let's see. Question 25. Horizontal gene transfer between bacterial cells. The best answer is going to be D, conjugation, transduction, or transformation are moving things between cells. 26, not possible according to the central dogma of molecular biology is the idea that environmental stress causes genes to mutate to resist the stress. Nah, probably not. It selects for mutations that already exist. All right. Let's see, 27. Which one of the following statements applies to conjugation? And that's where we have the interaction of two bacteria, and we have the F plus requirement of one bacterium to establish the bridge, and then an F minus bacteria is able to pick that up, and then in fact can become an F plus. Let's see, with the little micro essays. If you were close, you got everything. So purpose of the feedback inhibition, this is allowing a sort of a thermostat type control system in which you can allow a product to build up to a certain level and then begin to slow up its production. We need enzymes. These are critical because they reduce the activation energy of a reaction. They speed up chemical reactions. The role of NADH in respiration. NADH is a reduced electron acceptor. And so it is the molecule that is taking electrons as the food molecule is being oxidized. And then NADH is dumping its electrons into the electron transport system. Fermentation, it's better than nothing. You're able to get ATP, a few molecules, but you don't get as much ATP. It is not as efficient. So we don't typically carry out fermentation much. I mean, you do it when you're exercising really hard and you begin to generate lactic acid, but you can't maintain that for more than probably, a few moments, actually. So let's see. Advantages and disadvantages of sterilizing with steam heat. Steam heat is a really good way of killing stuff. The disadvantage is that there are a lot of things that can't handle being sterilized that way. There are some plastics that melt. There are going to be all kinds of proteins that are going to denature. You steam heat eggs, guess what happens? You make hard-boiled eggs. All right. Differences between each of these chemicals: sterilizing agents, disinfectants, antiseptics, and antibiotics. And this is the degree of reduction of pathogenic material or living stuff. Sterilizing, as the name says, is dead. Disinfectants are going to be doing a fairly harsh surface. Antiseptics are not nearly as harsh, and so that you can use them actually on the skin. Antibiotics are specifically targeting bacteria metabolically. So, two roles of plasmids in antibiotic resistance. The antibiotic resistance gene can be found on the plasmid. The plasmid then can be transformed from one bacterium to another. It's a way of transferring this stuff. Question 8, relationship between mutations and antibiotic resistance. Mutations generate antibiotic resistance. Antibiotics select for those mutations. Two ways a bacterium can become drug resistant. It can become drug resistant through a plasmid. It can be drug resistant through its own natural mutation, or through things like conjugation, transduction as well. Let's see, the difference between vertical versus horizontal. Vertical is the parent to offspring. Horizontal is within individuals in a community. All right, that's the exam. Exam 3 is about to drop. One thing, if you're listening here at this point in the conversation, Moodle is really miserable at these kinds of exams. And so it takes a while to kind of tease through them and to grade them. One thing that can really help is making sure you put your name on the doggone exam. If you don't put your name on the exam, it really becomes tedious because we have to go back and cross-reference the file to the individual exam and type the thing back in. And no, put your name on the exam. All right. On Wednesday, we're going to get into viruses that are involved in the blood, things like HIV, hemorrhagic fever, Ebola. Some pretty gruesome pictures. And I thank you for your participation. The article summary is the assignments in the works. We'll get it to you, but it won't be due until the week after Thanksgiving, so you don't have to rush about that. And that's all I had to say about that. All right, thanks, guys, and I'll talk with you on Wednesday. We'll see you. Bye.